works perfectly.
Okay.
Yep. You are good to go.
Awesome. Hello. Can everyone hear me on the internet? Yeah, my name is Lea Waller. I'm a PhD student in Berlin at Charité University Medicine. And I'm doing my PhD there on meta-analysis of fMRI data. And so when I set out, we really noticed we needed some kind of technology to do this. And so that's why we developed HALFpipe, which I will be talking about today. This is actually a talk I gave yesterday as part of the educational courses. So it's actually meant kind of that you might want to follow along. So I'm just gonna so you might want to follow along, you might not want to, but I'm just gonna leave this up for like 10 seconds in case you want to download it. There's also like an OSF link, but I don't know if I can show this right now. But yeah, if you screenshot this slide, you can get everything you need to start, and all of it is also in the GitHub in terms of these links for the downloads and so on. So let's get started. So in this talk, I want to talk a bit about what is HALFpipe, why did we develop it? Actually, should I take off my mask? Is that going to improve audio?
Yeah, go for it.
Okay. And talk about the framework where we developed it, what we want to accomplish with it. I'm going to then give a quick live demonstration with the example data set. So for the educational course, we decided to use this Amsterdam Open imaging cohort, which I have on my laptop, so we can like kind of play around with that a bit. And then finally, I will talk a bit about what to do with the outputs, what are kind of the use cases for when to use HALFpipe. And yeah, that's that. So let's jump right in. So you're probably, if you're listening to this talk, you might be planning your own neuroimaging study, right? You might have a hypothesis you want to test or a research question. You might want to, for example, compare groups. You might also want to do machine learning to kind of see if there's some kind of structure or information in your data. Or you might want to, like I am doing for my PhD, combine and compare data sets to see kind of if there's some overall structure to this data. You might also just have some data and want to explore it. That might also be very useful. And when I started out with neuroimaging, there are really a lot of great tutorials out there. So like really good papers that take you through all the steps, through all the different options that you have when doing processing. But in general, this can get very complex. And so you really need to do a lot of coding and debugging. There's a pretty steep learning curve in most cases. And one problem can also be that there's really a lot of options and choices when kind of designing your processing workflow. So I just tried to put all of these different options on this slide. So you have these different steps that will take you from your raw data to something you can actually use, to some kind of statistical maps or group statistics that are actually useful for answering a hypothesis. And as you can see, these are so many different steps. This actually gets really complicated in which steps you do and which parameters you choose for each step. So one tool that's really great that already exists is fMRI prep, and that will take care of kind of the basic steps for you and get you this minimally processed data that is really useful for getting started. But most neuroimaging studies will still have kind of another set of additional processing steps that will be done. And you will still also need to do some kind of data analysis to generate kind of maps on the individual level that are actually useful to answer research questions. And all of this needs to be implemented. So this is what HALFpipe does, and this is what where we come in. So what we do with HALFpipe is, of course, since we provide a kind of ready-made implementation, we take away a lot of flexibility. If you write your own script, that means you can kind of put in there whatever you want to put in there. And this has the benefit, flexibility can be good, but it can also be very bad, because in some cases, if every researcher does kind of the same thing, but a bit differently, you might get different results. And so the NARP study from 2020, they found, you probably will have heard of it, but just to summarize really briefly, different pipelines can lead to completely opposite judgments for whether a hypothesis is true or not, even if the researchers are using the exact same data. And this is really problematic. And so HALFpipe, by standardizing what you can do and by kind of giving you this kind of predefined pipeline, is one way to solve this problem and to get the same result independent of who specifically does the analysis. So how specifically are we doing this? We have kind of defined 5 steps that you can go through for your data. Number 1 is data input and configuration. So that means, of course, each data set that you'll be using, no matter how harmonized we are, is going to be a bit different. And so that means most data or some data is probably going to be in bits format, which is really great. And that makes it really easy to input your data. But if your data is not in bits format, that will also be kind of easy to do because we kind of provide this user interface where you can just specify, okay, what are the paths to my data? And once we have configured all of that and which types of analysis you want to do, HALFpipe will automatically do pre-processing using fMRI prep and calculate statistical maps for each participant. But now comes the second part where kind of things get a bit interactive and kind of human effort is needed because for quality control, we really believe you need to look at your data. And I think this is really important that kind of automated quality metrics can get you a part of the way, but it is really important at some point to just have a look at each participant to see how things look. So we decided to try to make this as kind of streamlined as possible by making a browser-based user interface that I will show later. So to try and make this as easy to use as possible. And finally, we provide a way to do group level statistics, which can also be quite useful. So I'm just going to show really quick how to set up HALFpipe and load in the example data set, which is called PIOP1, and it's from OpenNeural. So I'm now switching to my terminal. So I'm just going to show you what I have in my folder. I have my OpenNeural data set. I have my HALFpipe container file. I have some masks. So those are kind of useful for analyses like if I want to do a seed connectivity or an atlas-based connectivity, I can use these masks. I will talk about this in more detail in a bit. And I have some reports. So this is the QC thing. I will show this later. And I also have the working directory. So I mentioned I already did this yesterday. So I'm just going to delete the old working directory. But you can also reuse this. So it's actually not necessary to remove an old one. So maybe I'll just show what happens if you keep it. So to start HALFpipe, we're going to use Singularity. And I always like to Singularity run. I always like to use the option contain and clean env, which really makes sure that the container is isolated from the host system so that nothing kind of in terms of your computer's configuration can affect how the computation goes. And that's really good for reproducibility because no matter which operating system you have, the container is going to be run quite consistently. But I still need to give it access to my data. And I do that by binding a directory into the container. And I usually do that by just typing /, which is just like the entire file system. It's like the easiest way to do this because I'm just giving the container access to all of my files. And I'm giving it access at a specific path. And for HALFpipe, the convention is /ext. Now I just put the container. I'm using the latest version. We also provide releases that have numbered versions. And that might be good if you're doing kind of a more longer term project where it's really important that there's consistency with versions. But in general, the latest release will have kind of the fewest amount of bugs and will be the most advanced one that is available. So this will now take kind of a second to start up because, well, this container file is 5 GB in size. So it's kind of, it has all the software that is needed to run the entire processing, including fMRI prep, FreeSurfer, and so on. So it's a bit more involved. And so the first question I get now is specify working directory. And that's in my demo folder. So I'm just going to fill this in really quick. And I can do this so quick because I always get these suggestions down here that I can navigate with my arrow keys. And so then with enter, I can select one of these suggestions. But for example, there's only one suggestion. You can use tab to complete. And that's kind of simple. And since I still have my working directory from yesterday, I can just click run without modification if I want to keep that. But actually, because I want to show how it works, I'm going to start over at beginning, press enter. And so now I'm going to get questions about my data. And the first one is the data available in bits format? And if yes, that's super easy. I'm just going to show real quick what happens if I press no. When I press no, I'm going to be asked about each different type of kind of image file that might be, that will be needed for my analysis. I need to specify kind of the path to each of these, and then kind of, I'm going to show this real quick, work, demo, data set. And then, for example, if I have one folder for each subject, then I have to teach HALFpipe how to find all of the different folders. And I do that by, instead of the subject ID, I'm going to put kind of curly bracket subject, and it will kind of recognize this pattern that there's going to be sub, a dash, and then something else that is the subject ID. And that's how I can teach it to recognize a bunch of different files. And since we have a bits data set, I don't have to do this. So I can press Control C, which will take me back to the previous step. And I can just select yes and type in my data set. Okay, so this will take a second. I'll find the different types of data. And I'm actually just gonna show really quick what types of data HALFpipe supports, and you can load these in manually or automatically from a bits data set. And so it's anatomical functional field map data. And if you have task fMRI, then of course you will need some timing files. And I'll also go into this in a second. Kind of these additional files, I call them reference, also can be very important when you're calculating kind of statistics on your data. Okay, next question I get asked is, do slice timing? We usually recommend to say yes, but actually for this data set, we don't know what the slice order is, how they were acquired. So I actually have to say no, because I don't know the metadata for this data set. Next question is if I want to remove initial volumes from scans. And this can be important if kind of the bold signal is not equilibrated yet. And fMRI prep actually comes with this really great algorithm that will automatically detect this. But usually the scanner will just already have removed those volumes from the scan. So I can just also go with 0, and it's going to be fine for this data set. We don't have any field map files, so I'm just getting told that right here. And now I get to the question, specify first level features. And so of course I'm going to say yes, I do want to calculate some features, and I get these different types of features that I can choose. So task-based, seed-based connectivity, dual regression. So I'm just going to go through these in a bit and show what they are before we go back to the live demonstration. So what will happen now when we start processing is that we will run fMRI prep, and fMRI prep will run a bunch of these steps. One of them based on the kind of anatomical image, the high resolution image. It will correct the kind of intensity. It will do scar stripping. It will normalize the image. And for the bold image, it will do similar processing where it first generates a reference, corrects that for distortions, does some alignment and motion estimation. And the motion estimation is actually really important because that's how we get confound time series. So kind of time series that we can use later on to remove from the data, because this can really tell us something about signals that influence our data that we really don't want. So this is really an important output of fMRI prep. And the other thing that happens now, which I think is really great, is that kind of all the spatial information, so the head motion, the alignment, the distortion, and the spatial normalization, all of that is being combined in just one step. And then the functional data is resampled in just one step, and that makes it a lot faster and also reduces rounding error. So that's really useful. Then there are some kind of additional pre-processing steps that fMRI does not offer, but that are very common. So there's spatial smoothing, scaling. IC aroma is really good for kind of removing noise and keeping signal. So what happens is we run this ICA that independent component analysis will decompose our signal into different spatiotemporal components. And then the algorithm will kind of say, okay, these components are noise and these are signal. And then we can just kind of filter out those noise components. And so by default, we enable this. Temporal filtering, we also enable by default, but the specific parameters depends on what exactly we're actually calculating with the data. So that means kind of for smoothing, we have 6 millimeters, Grammy scaling, we scale everything to a mean of 10,000. We enable IC aroma, and temporal filtering, yeah, depends. And so those are kind of the additional steps that we suggest in HALFpipe, and they're all configurable. So, and then we go on to feature extraction. And this is what all will happen to your data before we actually will calculate the features that are being shown here in the user interface. And now I want to go a bit into what these specific ones mean and just talk about this really briefly. Oh, I'm sorry, I messed this up again. I always forget the order of this. So actually, I will first talk about a bit the confounds and what we're actually doing with these. So what is really important that we get these confound time series from fMRI prep, and we can kind of regress them out of the data. Like we are regressing out the noise components from IC aroma. We can remove kind of different parts of the signal. When we do temporal filtering, we also remove parts of the signal. And what can happen when we kind of in sequential steps remove parts of the signal is that conversely, and this is not really intuitive in like first impression. Oops. In the first impression, this is not intuitive, but I think this paper makes it really clear that if you do this, kind of just sequentially remove different parts of noise, you can actually reintroduce that noise back again. And that's really a big problem. And it's not actually that easy to solve, but in HALFpipe, we solve it for you. So you don't need to worry about this problem. And yeah, I really recommend checking out this paper. It explains kind of the problem really well with like some nice diagrams and arrows. Yeah, but I'm not going to go into any detail on that. And the other thing that we kind of really enable is that you can kind of compare different pipelines. So actually for the project I'm doing, we are comparing 3 different pipelines, and it actually does not add any processing time to the calculation, which is really good. So you can kind of specify different things you want to calculate, and it's going to be done in the most efficient way possible. So, okay, so now I can finally talk about the different things we can actually calculate with HALFpipe. The first thing is task-based activation. And so for that, we ask kind of the question, what is the difference between different conditions in a cognitive paradigm? So for example, which voxels in the brain correspond more or respond more to faces than geometric shapes, or more to reward than punishment, or for example, more to memory recall versus no memory recall, and so on. And when we ask this question, of course, we need some kind of information for when each of these conditions happens. And so we need event files that contain this information. And we also need kind of this information on the high-pass filter cutoff. The default for that is 125 seconds in HALFpipe, and that usually works. But for some paradigms, especially for kind of paradigms that have very slow repetitions, it can be important to kind of adjust this. So like to 1.5 times the slowest condition. And we use this to filter the design matrix for the regression, because in the end, what we are doing is we are doing a GLM, so a regression model, to regress the kind of events onto the functional data for each voxel. And one important aspect is that we remove these kind of low frequency drifts with this high pass filter. So it is really important to set this cutoff in the correct way, although in most cases the default will be fine. And finally, we also need kind of the contrasts of interest, so kind of which conditions do we actually want to compare and which ones are just in there kind of as a control or to filter out. And what we get out is we get out the GLM outputs. So the GLM outputs are these. So they're kind of, as you can see, if you're familiar with bits, they're a bit, these are just like the suffixes of the files. Before that, you will get a bunch of bits tags that actually identify the files, and you will get kind of the specific design matrix that was used, the specific contrast matrix that was used for the task-based design. And you can use these to kind of, judge that everything was done correctly. You will get the effect invariance maps, which is kind of also sometimes called COPE and VAR-COPE. You will get the t-statistic, which is derived from those. And from the t-statistic, you can do a hypothesis test and calculate a p-value, but we don't actually get a p-map, we get a z-map, and the z-map contains kind of, Z values that are kind of based on the normal distribution, which is much easier to work with than the t-statistic maps because you don't have the degrees of freedom to worry about. So this can be quite useful. But when we do group statistics, we actually want to use the raw effect and variance maps because that's a bit more sensitive. So, and you can find these outputs in the working directory. So there will be in the working directory a derivatives HALFpipe folder, and then for each participant, you will find these in there. One thing that is really cool is that based on the same GLM, we actually can also ask different questions about our data. We can, for example, look at connectivity. One of them is seed-based connectivity, and for this, we would ask the question, which voxels are correlated or show a correlated signal with a region of interest? And that is actually quite interesting because instead of our conditions, our event files, we're actually now using data derived from the brain as regressors. And so to find out kind of which regions show this correlated signal. And so what we need for this is a region of interest mask, so the seed region, and then we will get out, again, the same GLM outputs. We can also kind of do this in a bit more advanced way where we look at which voxels are associated with a specific brain network in dual regression. So in this case, we will have network templates, so a set of those. And in dual regression, we are assuming that our set of brain networks actually completely kind of, or describes in a way the different networks that our data is generated from. So in this case, we will kind of get kind of the specific contribution of each voxel to each brain network and can judge these again as GLM outputs. So it's a bit, so in this case, we have just one region. In this case, we have kind of different brain networks. So it's very related, but also a bit different. Finally, we can also look at atlas-based connectivity, where we look at pairs of brain regions that have correlated signals. This is based on a brain atlas, and we will get kind of a correlation matrix out of this, and also some other information. Finally, we also have these kind of more basal measures. They're very interesting for resting state, and also to just like explore your data. One of them is regional homogeneity, so which voxels have signals that are correlated to their neighborhood of voxels. And the other one is the fractional amplitude of low frequency fluctuations. So like how much compared to other voxels does this voxel have in terms of low frequency content. So these can also be interesting. And so when I go now here in HALFpipe, and I can, again, with my arrow keys, kind of select, and I can, for example, select a task-based feature that I want to do. And now I have to, when I press enter, have to specify a name for this feature. And I'm just gonna, I can specify anything. It's just gonna go into my output file name as a tag. So I can, for example, call this the OSR 2022 feature, just for fun, because we are in the open science room, and press enter, and then my output file is going to be named that way. And so, for example, if I want to work with the phases task, then I can just deselect all the other tasks with my spacebar. So these are kind of toggle buttons, and spacebar selects or deselects, and my arrow keys can again navigate between the different options. So I'm going to confirm with enter. Now you can see kind of which conditions were loaded from the bits data set. If this is not a bits data set, I have to load them manually, but that's also not a big deal. And I want to add all of those to my model, and I'm going to create a contrast now, which is phases. So everything has to have a name, right? Contrast phases versus baseline is my contrast. And then I'm just going to give a weight of 1 to all of these, which is basically just comparing them to the baseline in the model. Press enter, and I don't want to specify any more contrasts. And now I will just be asked about all the different things we already talked about. So the temporal filter for the design matrix, 125. I'm going to go with the default, which is good for this task. Next one is apply smoothing. I think I went over this quite quickly, but it's 6 millimeters is the default, and we can just go with that. It's sensible. It works for most paradigms and settings. I will now be told that Granby scaling will be applied with a mean of 10,000, which is sensible. We don't really have it in the user interface that you can edit this, but you can always edit the configuration file to set this to a different value. And now I'm also told temporal filtering will be applied using a Gaussian weighted filter. And I always take care that this one matches this filter up here. So this is up here is the filter for the design matrix. And this is the filter that we apply to the image, to the data. And again, I'm going to press enter. So the last question I get is whether I want to remove confounds. And by default, we have selected IC aroma here. But we can also deselect this again with the spacebar and, for example, select motion parameters. And then these will be added to my design matrix for my GLM. But I'm just going to go with the default because I think it's a sensible default. I'm going to press enter. So now I get prompted if I want to add another first level feature. So for example, now I could add another task-based model, but with different settings, maybe with a different smoothing or a different filter or different confounds, so that I can kind of make sure that my results are robust across different settings that I put into there. But I'm actually just going to set up a different feature type, which is seed-based connectivity, just to show how this works. And I'm going to go with the default name, which in this case is seed core. So the questions are kind of similar between different features. I'm going to press enter. And now I can specify which images to use. And for this, I'm going to go with the resting state because that makes sense for functional connectivity. I'm going to press enter. And now I need to tell it which seed mask I want to use. So which brain region is it that I'm going to look for, kind of what is correlated to that brain region. And I actually can go again to my work demos. I'm always using the tab key to kind of complete real quick. And I made this mask directory. And there's a lot of different masks that we generated in here. And I'm actually today interested in the posterior cingulate cortex. So I'm just going to select this mask. I'm just going to press enter to select it. And confirm again. And now I again need to give the seed a name. I'm going to call it PCC. That's again going to go into my output file name as a bids tag so that it's kind of every output file is identified. So finally, I will get asked for this seed, kind of what is the template space that it's in. And actually, this is because there's two different versions of the MNI space, version 6 and version 2009. And this one is in 2009 space, which is correct. So I just confirmed this. I can also now add more seed images, seed masks if I want to. But I can also just confirm with enter. And now we get to one important option, which is the minimum seed coverage. So depending how the acquisition volume was set in your data set, sometimes, especially for regions at the top or at the bottom of the brain, you will not have very good coverage of those regions for some participants. For example, if they have a very big brain or if there was some kind of user error. And so, of course, in this case, we don't want to have kind of artifacts. We don't want to have a bad map that doesn't make sense, that doesn't help us answer our research question. So what this says, if this is 0, that means we don't exclude anything. If this is 1, we will only calculate seed connectivity if every single voxel in the region of interest was acquired for that participant. If this is 0.8, we want at least 80% of the voxels to be acquired. And this is, I think, a sensible default. So I'm just going to confirm that. Now I am going to get the same settings as earlier for smoothing, temporal filter, and I'm just going to go with the defaults, confounds, and so on. And now I could add more features. I could add some dual regression. I could add more seed connectivities, more task analyses, and so on. And when I'm done, I just say no here. I can now also output a preprocessed image. And if I say yes, then I will be asked the same questions about which preprocessing settings I might want to do. I'm going to also select no. And now I can get asked if I want to specify a group level model. And in this case, I might get asked for kind of my variables that I want to add. So I can load in a spreadsheet with my sample characteristics like age or gender or patient group. I can choose to do subgroup analyses or specify custom contrasts and so on. So I'm not going to go into detail with this for time reasons. And I'm also going to select no. And so now I'm going to get a bunch of kind of like warning messages about my data because I have physiological parameters in here, and HALFpipe does not support these yet. So this is fine. And now fMRI prep already has started and is processing my data. So I'm just going to cancel this so that my laptop battery does not get drained and go back to the slides. So for the final part of this demonstration, I just want to really quickly show kind of what will be generated for quality assessment. So in the working directory, once HALFpipe has finished, you will find a reports directory. And in that reports directory, you will find an index.html file. So it's just going to be named index.html. You can just open that in any browser. And that's going to show you kind of this dashboard in the first view. And you will have on top kind of the processing status. And as you can see, all of my participants completed with success and no errors. And now that I've seen that, I can minimize this panel. The next panel is kind of the ratings that I've done for the images. So because you have to look at images and say, are they good or bad? And I've been lazy. I've only rated 4 images so far. So I'm also going to minimize that. And on the bottom now, I can see some histograms for my image quality metrics. And I can hover over the points, and I can also see maybe what went wrong with the different participants. For example, oh my God, this participant has a lot of motion. Maybe I can click on here and see what went wrong. Or and that takes me to, for example, the confounds plot or carpet plot. And so here I can then judge. I have like the frame-wise displacement here as a time series. And now I can kind of judge how are my voxel signals affected by the motion spikes. And I see things have been affected quite a bit by the motion spikes, right? So the blue one are the gray matter voxels. And we can really see there's lots of structure in here that shouldn't be there. So I would rate this image as bad. And so I can just click the frowny face or also use keyboard shortcuts that are documented in our manual to kind of do this in a faster way. And I always click to zoom and click to zoom to go back to the kind of tree view. I can also navigate here now to the other images for this participant. So I can look at the skull stripping, again, click to zoom. I can also go to the next image, which would be registration. So here I want to see that my template regions overlaid in kind of pink and orange and blue match my registered image in the background. I also get a TSNR, so temporal signal to noise ratio. Want to see that there is homogeneous brightness across the image and that there's no kind of obvious scanner artifacts in there. And I would say this image looks, yeah, more or less okay, but I'm not an expert on this, so I might rate it as uncertain so I can show it to my colleague and get their feedback. And then later on, make that into good or bad. So uncertain images will be included in group analysis by default. So, but you usually want to kind of make all your uncertains either good or bad before you do group analysis just to be safe. And now we're back at the confounds image. We also have the ICA Roma artifact removal. So this is just reports generated by fMRI prep. So here we see kind of what are the different ICA components that we got, and the green ones were classified as signal and the red ones as noise. And in my opinion, this one is kind of the hardest one to judge for quality control. So we usually don't use it that much unless the other images give us a reason to look into this one. But it's a lot of different components in most cases. So it's kind of hard to take it all in unless you're really an expert on ICA. And then the last image you look at, so it's 6 images per participant, is the EPI registration. And so here again, we want to see that the overlay of the template matches the background in the back. And I think it matches very nicely. And so once you've completed all that, you can go to the menu, click export, and it's going to download a JSON file, so a structured file that can be loaded into HALFpipe again or loaded into your favorite scripts if you want to kind of write them around this framework. And that will just contain all the information, all the ratings, who to include and who to exclude for group analysis. Yeah, so I'm just going to skip this for time. Yeah, you can do kind of preprocessed images. I showed this really quick. You can look at the subject level maps, and you can also do group statistics with HALFpipe. And this enables you to do both mega-analysis and meta-analysis. HALFpipe has lots of pros. You have automated workflows, but this also means we don't support certain things yet, and that will come at some point. But yeah, it might not support your specific use case. Pro is you do not need to bits convert your data. We do that for you if you don't have it done yet. But we're not a full-fledged bits converter, and we definitely do not validate your bits data, right? So it's not going to solve everything for you. We have, I think this is a really great pro, that we have suggested values and validated default parameters for preprocessing. And when we support kind of all these standard analyses you might want to do for both resting state and task-based data. But of course, that means also we don't support non-standard analyses. So you still need to do that outside of HALFpipe. We have this wonderful interactive quality tool that I showed just now, but it also means we really want you to look at 6 images per participant, and that's going to take you at least a minute. And so that can get annoying really quick for large data sets. So it can be a bit of a con, but I think, in my opinion, it's actually a pro, because looking at your data really makes your results that much more valuable. We have a detailed manual that explains every step in a lot of detail, but of course, it may not cover like what you need for your specific use case yet. HALFpipe is open source and also supported by multiple Enigma Consortium working groups that are using it also for analysis. But it also means we have no dedicated development and tech support staff. So it means that we are all volunteers working on this, and we're really motivated to work on this and also help you to use the software and get started. But of course, we are also only humans, so I kind of want to manage expectations a bit. Really quick on technical requirements, you can run this on your local computer or also on cluster computers for bigger data sets in parallel on multiple nodes. It will take about 4 to 6 hours per subject, but yeah, it could be longer if you have very high resolution data. You will need at least 8 gigabytes of RAM, so that old laptop that you have lying around is not going to cut it. And you also will need at least 1 gigabyte of storage per subject, which can really add up really quick if you have large data sets. If you also store kind of intermediate files, that will use much more data on your hard disk, but might make it faster if you want to reprocess something at some point. So yeah, if I have convinced you with this very fast overview and demonstration of HALFpipe, then please check out our GitHub and the manual, and also check out the paper that we've published recently on kind of the theoretical framework and technical considerations when we developed HALFpipe. It's out in Human Brain Mapping. And of course, this was all a huge group effort. So I want to just thank everyone in Berlin and Amsterdam who helped with development, everyone in the Enigma Consortium working groups who kind of did all this testing and helped us kind of, helped with all the different aspects that come with creating the software for the coin stack, and our 61 team that will help us with some other integrations, and also the wonderful people who have all beta tested the software through so many iterations until it's finally working as well as it is now. So yeah, please contact us at enigma@charité.de if you have any questions or problems. And of course, on the GitHub page, actually, I respond more quickly to the GitHub page than to emails. So this one is also great. If you have any problems, there's like a discussion forum. So yeah, thank you. I'm happy to answer any questions. And that was it.
We have some questions in the chat. When do you expect to include physio info into the pipeline?
I actually don't know the specific timeline for that, but I hope to do this. So we actually have a project in our lab that will use this. So it's actually definitely on the timeline, and it's definitely going to be soon. But yeah, it's really hard to say with software development because there's lots of aspects that go into this. But we definitely thought about it. We definitely have a plan for implementing it. So I hope in the fall maybe that we have this in there.
Awesome. And then there is another question that says, this is very cool. Thanks for sharing, Eric Earl. There's a lot of interactivity in these terminal menus. How do you re-trigger this pipeline on an HPC to repeatedly run these things without all the interactivity?
Okay, so the user interface is actually just like an interface. So it actually, what it happens if I go into my working directory, what it generates is this file called spec.json, and that JSON actually contains all the information that I entered into the user interface. And this is actually what is going to run HALFpipe. So it's actually two parts, like the user interface and the pipeline are completely separate, and they're just connected through this JSON file. So then that's what's going to be used on the cluster. So you're going to kind of generate the configuration, and everything else is going to be able to run reproducibly based on that. Does this answer your question?
Yes.
Okay, sounds like it. Yes. Thank you for the chat messages. So that's very quick typing.
Does anybody else have any last questions in person or virtually before we end? A lot of claps and thanks on the chat.
Yeah, thank you so much for joining, even though it is dinner time. Well, it might not be dinner time from where you are joining, but yeah, very happy that this was possible. Yeah, thank you so much.